I got the chat going, so you can type things there. If you have audio or visual, you can bring that up if you want. And I'm here to answer questions and things like that. So let me
see, I saw.
All right, so anybody got any questions? You're not going to see my video because I don't have a webcam, but I can obviously talk. We've got the chat, and I can bring up on top of my screen if you need it, a PowerPoint or something to explain something if you need it. So if you got any questions, so I got one chat question. It says, what would happen to labs such as labs and lab exams? Labs will be shown on a YouTube channel, right, where we have the lectures, and lab exams will be similar to what we've done before. So there you go. All right, so I also have a question about explain tonic control. So what tonic control is looking at is one of Cannon's postulates, right? So remember, Cannon had 4 postulates for homeostasis. Let's see if I can remember off the top of my head. So the 4 postulates for Cannon, that the ANS plays a significant role. There's antagonistic action. It's the receptor that gives the action. And then the last one is the tonic control. Tonic control means that there's always some sort of level of activity in the communication routes. So whether it's a hormone that's in the blood, whether it is a series of action potentials in a nerve, that there's always some level of activity. Sometimes that level of activity isn't sufficient to cause a physiological event, but the tonic control means that there's always there. And so we've used examples like men and women have the same hormones, right? And one was on the PowerPoint is that women, we use HCG, human chorionic gonadotropin, to measure whether or not a female is pregnant. So those are those take-home pregnancy tests you get in the store. Men have HCG even though we'll never get pregnant. So there's always some level of activity there, and tonic control allows us to better tonic control allows us to better increase or decrease that variable. And so that's what tonic control means. Is that sufficient?
Can you hear me? Are we
Yeah, I can hear you.
Oh,
Yeah.
oh, okay. In class, you gave this example of the blood vessels, and you said that they were controlled by the they had no parasympathetic innervation. So that's why we needed tonic control, because there's only sympathetic controls. So to dilate the blood vessels, we needed to send less signals, but I didn't really understand what you meant by send less.
So, so a blood vessel has to be able to constrict and dilate to control blood flow. So it has to get wider and it has to get narrower. If we didn't have tonic activity and you didn't have action potentials continuously going to a blood vessel, once you got to a certain level and the action potentials were off, we couldn't dilate that blood vessel anymore. Right, so having the ability to dilate less or more is important in terms of tonic control. So many of our autonomic functions have tonic control. Same with sweat glands. Our sweat glands are always getting some input from the sympathetic nervous system. So there is always tonic control, because even though we don't necessarily feel how much we're sweating, or because we're sweating very little, there may be instances where we want to sweat even less. So having tonic control allows us to turn that down even more. So there's just always some level of activity there.
Okay.
Anything else?
No, for me, that was it. Thank you.
Anybody on the chat? What else? Come on.
Can you hear me?
I can hear you.
Can you give me the answers to the exam?
Well, in the old days, I'd say, yeah. You know, I give you the answers to all the multiple choice: A, B, C, and D. You just have to figure out what questions they are, but that's probably not what you're looking for.
I'm just kidding. I was just watching the YouTube videos. Is that you talking on there? Or is that your voice
This is me. Yeah, that's me. Why?
on there? Or you sound different?
That was me. That I did that one at like 2 a.m. or 3 a.m. on Thursday, so it was pretty late.
You must have had a couple beers, huh, when you're doing it?
No, I can't afford. I was too busy to do that.
I'm kidding. No, I have 2 more to watch, so I don't have too many questions, but I'm sure I will probably after I watch them. But I just wanted to hear what you guys are asking on here. Maybe you know, someone can ask good questions, and so I'm trying to listen to what you have to say.
All right. So, question any more?
Yeah, there's another question on there.
I see it. IPSP and EPSP and graded potentials. Okay, that's a good one. So graded potentials, let's kind of start from the beginning. Graded potentials are part of that local communication that we talked about, where in a graded potential, it's caused by either opening or closing a single gated ion channel. Okay, there are actually, let me do this. So there are actually 4 different types of ion channels that we worry about, and I can show this on the screen. There it is.
How do I look on the screen?
I haven't done anything yet.
Oh, sorry. Okay, I'm new to this. It's my first time.
Yeah, it was my first time too. I practiced once today, but I'm going to share this PowerPoint with you. And so now you should see it on the screen. Yeah? Yeah. So here's what's basically talking about the postsynaptic graded potentials. So there are 2 types of postsynaptic graded potentials. There's the IPSP, which is the inhibitory signal, and the EPSP, which is the excitatory stimulus. Okay. So going back to this action potential here, right? If you look at the resting membrane potential, you can see the rest. We know that it's about minus 70 millivolts, and what we want to do in order to trigger an action potential is we want to make that minus 70 get up to that threshold potential of minus 55. In order to do that, we have to have an excitatory stimulus, right? So that would be the EPSP. It's an EPSP because it takes us closer to threshold. So when we look at that, we have 4 ways to make an EPSP. Okay, the first 2 are pretty simple, and just kind of mentally think in your head that sodium and calcium, right, kind of go together. And so they both have, if you look over on the side of the drawing, higher concentration outside the cell. That's why you see them outside, right? And a low concentration inside. So if you look, you can see a tiny little Na plus and Ca plus 2 on the inside. And so if you open a channel for sodium will diffuse in down its gradient through a channel. We've talked about that before. Since sodium is a cation, it's positive, it's going to make the inside of the cell more positive. And as it makes the inside of the cell more positive, going back to this one, it's going to take that resting membrane potential and move it towards threshold. Okay, so that would be one way to make an EPSP. Now, the other way to do it through an open channel is calcium. Now, calcium is very similar to sodium. It's a high concentration outside. It's a cation. So when you open a channel, it'll diffuse in, and it diffuses in because it's a positive ion. It makes the cell more positive. So those are two ways to create EPSPs. There's actually two other ones, which are a little less intuitive, right? And the less intuitive part is, you kind of think about it this way, and we mentioned this before in class, but this is not a bad analogy. If you want to, save $1,000 so you can go on vacation when this stuff's all over and you actually go outside and interact with people again, right? Then what you want to do is you want to save money. Now, there's two ways to save money, right? One is to make more, and the other one is to spend less. And so the other way to make the EPSP is like the opposite way in terms of saving money. So what happens is we would have already a channel open for chloride. Okay, so if there's a channel open for chloride, chloride is similar to calcium and sodium in that it's a high concentration outside, so we can see it big outside. It's a low concentration inside. But when we open a channel for chloride, right, chloride moves in. Since it's an anion, it's going to make the cell more negative. So if I close that channel, you can kind of see a little X in the middle of the channel. If I close that channel, chloride stops coming in, which means that the membrane voltage is going to go up because you're not bringing chloride in anymore. The other way to do it is you could close a potassium channel. Potassium is that unique one, right? It's the most abundant intracellular cation. So it's a high concentration inside. So if we open a channel, it will move out and it'll make the cell more negative because we're removing positives from the inside. And so if you close that channel, then potassium stops leaving and your cell stops going negative. So to make an EPSP, that depolarizing stimulus towards threshold, you open a sodium or calcium channel or close a chloride or potassium channel. And an IPSP is just the opposite. So does that make sense? Anybody, everybody?
Really quick, IPSP, that's your, you did a little representation of that's saving money. And then the
Yes.
and then the other one is the making more money.
Yeah, you can kind of think of that way.
Okay.
All right, that makes sense?
Professor Hoffman,
Yes.
if you could do like a sentence just summarizing the difference between them one more time, are you able to do that?
Yeah, so an EPSP is an excitatory stimulus that drives us towards threshold. So it's known as depolarization. And it's caused by opening calcium or sodium channels or closing potassium or chloride channels. The IPSP, that's the inhibitory signal, right? That is a graded potential that, again, through single gated channels, it's going to drive us away from threshold. So that's hyperpolarizing, right? It goes farther away. So that's hyperpolarizing. And that's created by the opposite thing. So you open a chloride channel or a potassium channel or close a sodium channel or calcium channel. So that would be a summary of those really quick.
Thank you. Thank you.
Thank you. I had a question just to confirm. So the membrane threshold is negative 70 millivolts, and the threshold is negative 55 millivolts.
So the resting, the resting membrane potential is minus 70.
Okay.
Right, and the threshold is minus 55 for an action potential. Okay.
Okay. Got it.
So to get an action potential, right, and this is kind of this is actually a great question, a great segue. Graded potentials, I said it a couple of times, right? Get in your head, there's single gated channels always, right? Action potential, double gated channel always. So it gets a little confusing because we have a single gated channel for sodium, right? That makes the IPSP or EPSP, depending where you open or close it. We also have a double gated sodium channel for the action potential, right? And those double gated sodium channels, and I think we can understand this because we've talked about it before, are voltage regulated channels, which means when they reach a certain voltage, they open, and that certain voltage is minus 55. That's our threshold voltage. So if you're, trying to learn stuff, then I got it somewhere.
What was single gated again? I'm sorry.
All the graded channels, any of the graded stuff that we
Oh.
talked about are single gates. So
Okay.
remember, for us, action potential double gate, right? And then everything else single gate. It's probably the best way
Okay.
to think.
Okay, thank you.
Oh, that helps. Yeah, action potential double gated.
I'm trying to having trouble hearing that one.
Okay.
All right, other questions?
I do have another one.
Okay, don't, yeah, it's fine. That's what we're here for.
Okay, so for part one, the one that we do at home, the COVID edition, where the number one, where we have to assume that the number of ATP produced is 38 total. So there's 38 in both. There's glycolysis. I mean the malate
aspartate shuttle.
And the glycerol phosphate shuttle. So we can explain either one.
No.
Or
so what you have to do, right, is pull out the schematic, right, that we have, and the 38 total tells you which one to use. So if you remember, the one we did in class was 40 total. Right? And so what shuttle do we use, remember? You just mentioned it.
I think I'm looking at the wrong. I printed out. I'm looking at the wrong. Let me look at the one from class.
Malate aspartate.
Yeah, so we talked about malate aspartate in class. And how many total ATP did it give us?
Hold on. Wasn't it 38?
So malate
aspartate. No. That's my. Sorry, it's 40. The other one, 38, is the glycerol phosphate shuttle.
And so what does the question ask for?
I'm not gonna lie. I didn't really read the whole question.
You just read the question to me. What was it asked for?
Oh, assume that the number of ATP produced is 38 total in this case.
So now match it up with what you just said before.
Which is the glycerol phosphate shuttle.
There you go.
Okay. Oh, I hope I do good on this exam.
Yeah, I hope everybody does okay.
Sorry.
You guys did a lot better on the lab quiz than I thought you would, so that was good.
Same. I mean, I thought I was gonna fail it.
You guys did pretty well. So
I started a new job, so I'm really not on I'm not sheltered in. I'm really still doing a lot.
Yeah.
I actually think passed by a COVID patient because the security told me to back away, and I'm like, why? And he's like, back up 6 feet. I was like, oh shoot, this patient is coming from ICU. This is kind of scary.
It is kind of scary.
Yeah. Really
but that's why we're doing this, so you don't have 50 of them, right?
Yeah, that's true.
Exactly, yeah. So Professor Hoffman, on our notes, remember how we did all the breakdown of numbers? Under mine, I have total. It says total, and then that's when we have 2 numbers. So what is that total? Like we had a glycolysis and all the numbers, activation, all the numbers, Krebs cycle, all the numbers, and total. And total is where I do see 34 over 32 for ATP indirect and 40 over 30 ATP total.
Yeah, so the question asks, right, if you have 38 total ATP, how'd we get it, basically, right? So you just mentioned, yeah, we get so many in glycolysis, we get so many in activation, we get so many in Krebs, and this is how we add it all up. That's what you're going to explain to me.
Okay.
Is total the subtraction of the cost from the net?
No, that would be net, right? So total is how many ATP you get irregardless of the net. So find the total column, right? and go down to the bottom and see what the biggest number is. That's the 40 or the 38.
So I have like a total like category. It literally says total, then it says like ATP used 2, ATP direct 6, NADH.
So you're looking at the total of the total.
Oh,
So the bottom row is the total, but find the column that says total ATP. And where those two total meets, that's the number you're looking at.
got it. Okay.
That makes sense to everybody?
I think so.
I think I need to see someone's notes. I hope I wrote this right.
Renee, I have the
I really
chart.
hope I wrote this right, Sarah.
What?
I hope I wrote this right.
Let's see.
Oh, okay. Another question, Professor.
Yes.
For the multiple choice or those 3 short answer questions,
Mm-hmm.
or do you think potentially we're gonna have like questions about metabolism, like this kind of stuff, like the ATP total or net, or are we just keeping this just for this separate assignment or separate part, I guess?
Well, if you looked at the announcement I put out with it, I said doing this in advance or looking at it in advance might help you study for the exam.
Yeah,
Right?
you did say that.
So obviously I'm not going to ask the same question because I already got that question asked, right?
Yes.
But we saw some of them already with the questions about how many, right? How many ATP do we get from this? How many NADH and all those different things? There's a number of those.
Mm-hmm.
There's probably 4 or 5 of those questions on the exam.
Can you do a quick brief like summary on Krebs cycle of like how we can just distinguish if you're talking about the whole entire thing or just one? Remember how you said?
Okay, hold on. That's a good question. We also had another question I don't want to forget about with the current leaks, but we'll talk about that. To be honest, the current leaks aren't very essential at all, but we will talk about conduction and kind of make sure everybody understands that. Let's see. Hold on. Well, you can talk, but I'm.
No, okay. Just the Krebs cycle in particular.
Yeah, I'm looking for what's the best material to get to see what I'm talking about here. So.
I like that you can pull up the stuff so we can see it live. That's cool.
Yeah, I am trying to find. That wasn't what I wanted to do. I want to share the screen, but I don't see in my thing what I'm looking for. I don't know how well sharing my screen does with. It doesn't seem to work very well with PDF files. I don't know why.
Oh, are you able to do like a whiteboard?
Yeah, but the whiteboard is I tried drawing on it with the mouse. It just doesn't work, and I don't have a screen I can actually draw on. Not that it would be much better, but
it is hard with the mouse. Yeah.
I got other options. I'm not at options yet. All right, so I have that here.
Would you say that's the most tricky one? Is the Krebs cycle? I guess out of them all, I guess.
For some people, maybe. That's not what I want to share. You can see that, right? That's the announcement of this. Okay. All right. Well, let's see if I can see that. I should be able to see the group problem thing. I don't know why I can't. is finally. All right, so this is what, this is what I was looking for. Everybody can see that?
Uh-huh.
All right, so here's the, looking at the glucose, right, using the malate aspartate shuttle. And in this case, right, so this is our group problem. We make 40 ATP total. All right, so when the question was asking about one turn or whatever, right, and this ain't showing exactly, I think it got kind of messed up when they're showing it, but I'll use this side. So if you look at the left side where you can see after glycolysis, when the pyruvate split and then went into the mitochondria, we'll just follow this left side. It becomes acetyl coenzyme A, so that's the one activation step, right? And then that one acetyl coenzyme A binds with one oxaloacetate. That makes one citric acid. So this one, look how fancy, acetyl coenzyme A, right? Goes there, right? And makes one citric acid. And then it goes through a series of steps and becomes oxaloacetate again. This is what we get. So this is one turn. And if we look at the other side, right, this is the other turn. So if you add these together, that's what you would get down here. Oh, I gotta get out of this.
So like adding those would be like the 2 ATP plus like 6 NADH. Like that's what you mean?
Yep.
Okay.
So 2 ATP, 6 NADH, 2 FEDH2, and 4 CO2s, right? Even though partially covered. So let me do this again. Get those out of our way. And so if you look at the bottom of this, right, we can see for the Krebs cycle, it's exactly what you said. We got 2 ATP, 6 NADH, 2 FADH2s. Then we do that math, right? 18 plus 4, because I get 18. 6 times 3 is 18. 2 times 2 FADH2 is 4. 18 plus 4 is 22. So that's the indirect. And then looking at the total, right, is 24 for Krebs. Net is 24 because it's subtract out 0. And then when we were talking about the total, then you find the total at the bottom and the ATP total here, right? So if I go down to there and I go across to there, that's the number I'm looking for, 40 total. So, and then the malate aspartate, right? That's 40. The glycerol phosphate, right? What would happen is the NADH would become FADH2, right? So, and then that would make that 4 instead of 6, and then, well, it's hard to do with the mouse, 8 instead of 10, and then 6, and that's how come when we add down, 8 plus 6 is 14, and 24 is 38, so the total would be 38 if we use the other shuttle. Does that make sense?
That does. So, and then, so just for like terminology or, you know, wording, when you're asking, are you saying of 2 cycles or 2 turns? How do I know, like?
Well, if I said, I wouldn't, if I said 2 cycles and you just add it together, right? If I said 1 cycle 1, if I said for a glucose molecule, how many cycles am I talking about?
You do
1.
both of them.
If I said for a glucose, right? You do both of them, right? As most of you said, because 1 glucose gives me how many pyruvates?
2.
2.
Yeah,
2.
2.
Yeah, 2.
So then you get 2 cycles, right? Make sense?
Yes.
Can you give us a trickier one then? Like not saying glucose?
Well, we wouldn't do that without glucose, right? Because you got to start with glycolysis. The only thing going to go glycolysis is glucose.
Sorry, I mean, like if like a question were to ask how many, what's the total ATP from?
Like what? Okay, so let's go. We didn't really do it yet, right? What's the indirect ATP from 1 turn of the Krebs cycle?
The indirect.
Indirect ATP from 1 turn of the Krebs cycle.
Okay, that's when I get confused. So it's not 22, right? Because it's 1 turn. Is it considered 2 turns when that's the full thing?
Yes. So 2 turns are for each one of those sides, right? So 1 turn is just what do we get? So so first of all, we're just doing one side. That's one turn.
Uh-huh.
Okay. What's indirect ATP? How am I going to count that up? How much ATP I get from NADH and FADH2? So one turn of the Krebs cycle, how many, let's do NADH first. How many NADHs do I have for one turn of the Krebs cycle?
3, 2.
Three, good. 3.
Oh, yeah,
How
'cause
many
NAD
FADH2s?
1.
2. Oh,
FADH2. How many do we have?
1. Oh, in the Krebs cycle, just 1.
One turn,
One
right?
Krebs.
yeah.
Was 1. Okay. Now, how much is that FADH2 worth when we cash it in for ATP?
2.
2. So hold that off
ATP.
instead. Side of your head, 2.
Okay.
How many NADHs do we have? 3
6.
times
Oh,
3 ATP is
3, 9, sorry.
9,
That's
and 9
9.
plus 2 is
11.
11. So one turn of the Krebs cycle indirectly would give me 11 ATP.
Oh, you don't count the actual ATP?
No,
The
because
one
that's
ATP?
direct, right? So
Sorry, I wasn't here when you did that lecture. Remember, I would
no,
take
that's
a
fine.
flight.
That's fine.
So
No,
I'm like,
don't
okay.
apologize. That's fine.
Okay.
All right. So the other side of that question would be, right, or another side of it would be, how much direct ATP do you get in one turn of the Krebs cycle?
1.
1.
1.
Very nice. Okay, don't fall for it. Right?
Oh,
So
shit.
How many ATP do you get through? How many ATP do you get through substrate level phosphorylation
Oh my gosh.
in one turn of the Krebs cycle?
Wait, say that again.
ATP,
2,
substrate
right?
level phosphorylation, one turn of the Krebs cycle. So substrate level phosphorylation is the exact same as what?
Oh, isn't that the
direct?
same as?
Direct, yes, very
Yeah,
nice.
1. So it'd be
Oh.
1.
It'd be 1 again, exact same question.
Oh my gosh.
All right. So
This is
those are
too
the examples of some of the harder ones I could ask.
Okay, so isn't there like 3 or 4 different names of the same thing?
Yes.
What's
Okay,
the
I think
reason?
I wrote them. Hold on, I think I really wrote these things. Okay, so wait, we're talking about the Krebs cycle, right? Hold
Well,
on.
substrate level phosphorylation is the same as ATP direct. But
Yes.
why is there's 2 different, like, why do we reference them in 2 different names?
Because
It's science and they want to make us.
Yeah,
to
exactly.
some extent, right? I mean, you know, sometimes it's one's maybe a better description than the other, but yeah, to make things harder sometimes, right? Well,
So
so let's finish the other side,
Oh yeah.
okay? So on the other side, indirect would also be what?
1. Oh no,
No,
indirect would be
talk about
11.
the other names.
11.
Electron
Oh.
transport, nice, and oxidative
Malate.
phosphorylation.
Oh my gosh.
Yeah.
Oxidative.
Okay.
Wait.
Can we repeat that real quick one more time?
So, ready? Direct is substrate level phosphorylation. They're interchangeable, same thing. And then indirect is the same as oxidative phosphorylation and the electron transport chain.
Thank you.
Thank you. Substrate phosphorylation is the same
Substrate
as
level
direct.
phosphorylation.
Okay.
Yeah.
Oh, substrate level is for direct.
So at any point, if you ask us, like, what is the amount or like the yield of the electron transport train, we have to know it's the indirect ATP
It'd be
that we're
the
talking.
indirect, exactly.
And you'll say if it's like one turn or both, right?
Yeah, I'll say for like a glucose molecule or for one turn of the Krebs cycle or something like that.
Okay, and then for a glucose molecule, the ATP direct would be, wait, where am I at?
For a
6?
glucose molecule, let's do that. The total ATP direct for a glucose molecule.
Total ATP direct.
So do you count the ones in the cytosol too, or no? I don't know.
Yeah, so it's just the total, right?
Oh.
So we're talking about all of them now. Yeah.
So there's 2, and then it breaks up into 4. So you count those 2?
Yeah, so you got
4,
you
6.
Got, and stop there. 6.
So 6. Okay, 6 in a cytosol.
Does that make sense? I can share that. All right.
So that would also be called direct too. The ones that are in the cytosol, is that also called direct ATP, or no?
Yes.
It is, right? Okay.
Yeah. So again, looking at this screen, right?
Uh-huh.
So direct ATP, right, would be this column right here. And so you get 4 from glycolysis, 2 from Krebs, so that gives me 6 total. All right. Now you made another nice one,
Oh.
right? So, and this is kind of, so how many direct ATP do we get in glycolysis?
Cool.
So you
Direct
just look at
What?
you just look at this column.
4.
In glycolysis would be 4,
4.
right?
Yeah.
So
Yeah,
if I
yeah.
said how perfect, right? How many do you get through substrate level phosphorylation?
0.
No, because remember,
One.
direct
Oh,
is
4.
Well, two.
no direct,
4.
right? Is the same as substrate. So if it's 4 for direct, it's also
4 for substrate.
4 for substrate. Perfect. Okay. Now, if I said, how many do you make in the cytosol for a glucose molecule, right? That's the only place, this blue, right, is the only place where we have ATP made in the cytosol. Does that make sense?
So just glycolysis.
Well, and just glycolysis, but what about this NADH here? What about this NADH right here? Why don't we count that NADH? Those 2 there. Because where do we actually make the ATP?
In the middle.
In the mitochondria, right?
Yeah.
So that's why if I said, how many in the cytosol do you get? The answer would be 4.
Oh, you don't count the 2 that come right off the glucose. You count one right before it goes to pyruvate. That's the 2 that you want, the 4 that you want to count.
Yeah, so the 2 on the one side, 2 on the other.
What do you call that? Because the one before that is direct ATP, the 2, and then what do you call the other 2 that split up? That's not direct anymore, right?
Well, the are you talking about the top here?
Yes.
Or
So that's direct, right? The one you're looking, that's direct, right?
this one, that one right there?
Yes.
Those are putting in, right? They're not coming out. So those are used.
Oh, used. See,
So those are just the used one. So we're not getting them out.
okay.
So they're not direct or indirect.
And
or
what
anything
about
with
the
you.
next 2 that split up?
So if it says ATP, it means they were direct. Boom, they're done.
Like where's right before pyruvate, those 2 ATPs.
Yeah, so they're
Yeah.
direct. So
Oh, it's called direct. Okay.
those are direct.
Oh, it
And
is direct.
anytime,
Okay.
anytime you see ATP come out, so the arrow's coming out, that's direct.
Oh, coming out. Okay. Oh yeah, that was.
Notice the top arrow, that's going in. So those are the
tricky.
used. These are that's the yield. Matter of fact, if you look, every arrow, right, is coming out after that. So the only place the arrow goes in is at the beginning here for the
Oh,
yield.
okay, okay.
Right.
And we always seem to start with 2 ATP, I think, right? In every single.
And in glycolysis, always, no matter
Yeah.
what happens to it, you end up with that.
But like activation, we don't.
Yeah, can you go over activation real quick?
Cycle, we don't. Or no.
All right, so let's go back to activation, right? So the activation step is just once pyruvate has entered the mitochondria, it gets converted to acetyl coenzyme A. Okay, that's the activation step. And in that step, we make 1 NADH. That's eventually going to be worth 3 ATP. And we make 1 CO2 because we go from a 3-carbon pyruvate, right, to a 2-carbon acetyl coenzyme A. Since we have 2 pyruvates for 1 glucose molecule, this happens twice for the whole thing. Okay. So what's my indirect ATP for the activation of 1 pyruvate molecule? 1 pyruvate indirect ATP.
You're asking for ATP, the 3 of them, right? 3?
3 of them, right? Because I get 1 NADH, that gives me 3 ATP. What's my CO2 for 1 activation step?
1.
1, perfect. Okay. So for how many CO2 do I get in the activation step for a glucose molecule?
2.
2, because you count both sides, right?
Oh, yes.
Making sense a little bit?
So when you glucose in general, you total them. You add both sides together. Okay.
Yes, if you're doing a glucose, you total both sides. Perfect.
Yeah, that's helpful. That's really helpful.
Thank you so much
Okay,
for doing
so
this.
let's, couple other questions that came in. Let's go back to this. So one of the questions was asking, we'll go back to the chat for a second. Okay, so I'm going to talk about orthodromic and antidromic, and I'm also going to talk a little bit about the gradient, okay? So let's look first at the gradient, and to do that, I'm going to share this. Come on. All right, so all the gradient is talking about is there's two types of gradients, okay? There's a chemical gradient, and so if you look right here, this is supposed to represent like a fake cell. And if you look at potassium, okay, and I'll look at, here's potassium. Here's the concentration right here, 140 millimoles of potassium on the inside of the cell. And if you look at the outside, it's about 5 millimoles, okay? That's really close to what we do with human cells. Humans are 140 to 150 inside and 5 outside. So there's a chemical gradient from inside to outside, and that's all that refers to is what's that diffusion gradient, right? If we look on the other side, we see that we have 150 millimoles of potassium chloride here, right? We're just going to worry about sodium chloride on this side, sodium chloride. So on the right hypothetical cell, we have sodium. Now notice sodium's high on the outside, low on the inside, right? It's 150 to 15. So this is only about 10 to 1, as opposed to the potassium one that was 30 to 1. So there's also a chemical gradient here, but it's not as much because that concentration difference isn't as much. So that's all it's talking about, is the higher the concentration difference, the greater the chemical gradient will be. In the case of potassium, because it's high inside, its chemical gradient is out. And in the case of sodium, right, it's high on the outside, so its chemical gradient will be in. So its chemical gradient will be in. Now, there's also an electrical gradient. So let's go back and look at the potassium one. Potassium has a high concentration inside the cell. So notice the potassium are the little diamonds, right? So there's one little potassium, and there's another, and there's another. And notice there's a whole bunch of them inside in the tan color, and not very many in the blue color. So what's going to happen with potassium is potassium is going to diffuse out of the cell. As potassium diffuses to the other side, we're taking positive ions away from the inside of the cell and moving to the outside. And as we do that, it's just like your checking account. If you spent money, your checking account will become more negative. So the inside, that's what these little negatives here represent along the edge, that the inside of the cell is becoming more negative. As the inside of the cell becomes more negative, it gets harder to leave for potassium because it's a positively charged ion. And that's called the electrical gradient. And once that electrical gradient gets high enough, it says even though potassium has a chemical gradient to leave down its diffusion pathway, it says you can't leave because it's got that electrostatic attraction, and the pull by the electrical gradient is equal and opposite of that. And that's when we have the equilibrium potential. And that happens at minus 90 millivolts for potassium. So the electrical gradient is always going to be opposite the chemical gradient. For sodium, it's similar but a little bit different. Since sodium is a positively charged ion, as it moves into the cell, it becomes more positively charged. And so again, find my cursor. So sodium is represented as these little yellow dots, and so there's more on the outside than the inside. So sodium diffuses in because sodium is a positive ion. When it becomes into the cell, it becomes more and more positive. Okay, so sodium is moving down its chemical gradient into the cell. As it does that, the inside of the cell becomes more positive, and it starts to push back on sodium. So instead of having, like potassium had, that electrostatic attraction because the two opposite charges attracted each other, here it's electrostatic repulsion, that the sodium can't come in. Once there's a big enough positive charge inside the cell, the electrical gradient's high enough, basically, that it pushes back on sodium, says you can't come in anymore because they're both the same charge. And since they're the same charge, if you ever tried to put like two ends of the magnet together that are the same charge, they repel each other. So this is called electrostatic repulsion. So that's when sodium quits coming into the cell, and that happens at about 60 millivolts. We're going to round down to 60. And those are how we figure out the equilibrium potential.
So when it's equilibrium, it's even on both inside and outside. So would it be like, for example, 30 millivolts inside and 30 millivolts outside to make it 60?
No, it would just, what we do is we measure the inside compared to the outside. So the inside's going to have more positive ions at that point in time.
Okay.
And so that's where it's going to, so it's when there's no net movement of sodium into the cell. And it happens when that charge has to get a little bigger, okay? And when it gets a little bigger, then that's called the Nernst equation. And when it gets a little bigger, it prevents it from coming in. One of the points then kind of look at here, which I didn't mention, but if you look at the one for potassium on this side, so it's that minus 90 millivolts. Well, I kind of covered up the 90. And for sodium, it's plus 60. The reason why potassium has a bigger number is it's got that bigger concentration difference. And since it's got a bigger concentration difference, that gradient is bigger, and it's harder for the charge to hold on to potassium until it gets big. So the bigger the chemical gradient, the bigger the electrical gradient that needs to be to oppose it. And so the higher concentration differences are going to have bigger equilibrium potential. So that's why we said that sodium is plus 62, but we're just going to talk about plus 60, and potassium is minus 90. And so those are numbers that you should learn and should sort of memorize there. Clear that and clear all the drawings and stop sharing that for a second. So that make a little bit of sense?
I get the chemical, but the electrical, I don't know what. I have to know like when it stops. I mean, once it makes it equal. I don't know why I'm getting confused with that part.
You should know when it's equal, and just know the number, right? So memorize those. Plus 60 equilibrium potential for sodium, minus 90 equilibrium potential for potassium.
Okay.
All right. Before I forget, I had a couple other quick questions that people sent over. So what I'm going to do, I'm going to share the screen again on a different slide. So this is looking at the conduction stuff. And this is going to kind of answer two questions. Actually, maybe we could even answer a couple more. The slide before that talked about something called current leak. And current leak slows the conduction down. And basically, it really refers to the fact that in order to get the action potential to move down the axon, so on this one, you can kind of see that we have the depolarization, right, going down in the direction from left to right. And that's because we're going through, and they don't show it here, but at the node, right, there are double gated sodium channels. So they show the channels, they don't show the double gates, but they show the double gated sodium channel. And then they show the axons, right, with the myelin. So if you look where there's myelin, notice there's no double gated channels. So the only place that has the channels are the nodes, right? So those nodes of Ranvier. And what happens is that there's current leak here. It just means we have to open more channels is the best way to think about it. While here, where there's the myelin sheath, we don't have to open any channels. So we get to literally skip that part. You actually do it on the inside, but we skip that part and we jump to the next node. And so the having the myelin basically prevents current leak. What it really says, right, is you have to open less channels so it takes less time to go from one end to another. So basically the myelin helps to prevent current leak by insulating. But that's, you don't have to know that much. Just the whole thing about that is know that myelin speeds up conduction because the nodes are the only place we exchange the ions, right? So here's the channels and here's the channels right here, but where there's the myelin, notice there's no channels, okay? The other thing that's kind of important then is we're gonna go away. It's probably not a bad one to look at. So the other question I had was about orthodromic conduction, okay? Now, orthodromic is just a fancy scientific name to say conduction in the correct direction. And in every case, right, conduction should start here, right where the cell body meets the axon, right at the axon hillock, right? And right there, the axon hillock, which is known as the trigger zone functionally, has the double-gated sodium channels. And I have to open that first double-gated sodium channel here. And when I do, then because it's a double-gated channel, it has 4 channel configurations. Remember, so the double-gated channels have 4, the single-gated are just 2, either open or closed. But the 4 gates, you have to worry about what the configuration is, but you know it has multiple configurations. So once we get the action potential to fire, then the double-gated channel goes in a very specific orientation that it can't get excited anymore. Remember, we called that the absolute refractory period. And what it does is it prevents the electricity from going backwards and makes it go forward down the axon. The direction we want it to go is called orthodromic conduction. So orthodromic means the correct direction. That's from the cell body to the synaptic end bulb. If it was going in the opposite direction, that would be called antidromic. And antidromic would be bad because then it would disrupt any information coming the other way. Plus, there's no signal receivers at that end for it. So we always want to make sure that we go in orthodromic conduction. And I don't want to confuse you with that one.
When does it go backwards? Like in what cases does it go backwards?
The only time it would go backwards potentially is if we had an electrical synapse, okay? And so electrical synapses, notice here's an example that I just put up here. When you have an electrical synapse, what happens is that electricity can be passed from either side in either direction. And so electricity, notice that we've got these double-headed arrows on both sides. That means electricity going either direction. For a normal chemical synapse, it can only go in one direction. And the reason why, I'm going to clear it to move to this one. The reason why is that in this instance, this is a chemical synapse, and notice that the excitable channels are all on the postsynaptic membrane that are going to carry the information across the synapse. So having a chemical synapse makes the signal go in the correct direction. That's called orthodromic. If it went in the wrong direction, it would be antidromic. So you know, functionally, we don't have that very much, except at the very few chemical synapses that we have. And so that's the problem. We don't have a whole lot of electrical synapses because they're bidirectional. And since they can travel in the wrong direction, we don't have the proper control. So our body gives up a little bit to have a chemical synapse, which is a little bit slower, but it's not that much slow. The average synaptic delay, you don't have to know this, of a chemical synapse is about 1/2 millisecond, so 1/2000th of a second. But it assures that the action potential in this case, right, comes down the axon, right, and then goes across and to the other side. So that's the advantage of having this chemical synapse. It's a little slower, but we give up a little bit of speed for a lot of control. And so, you know, I always, you know, in class, I would use this argument why chemical synapses aren't that good. Maybe you want to, let's say, maybe you want to drive a car that's, you know, really, really fast. So you go, okay, you're going to buy a car, and one goes 200 miles an hour and doesn't have a steering wheel, and the other one goes 195 miles an hour and has a steering wheel. You know, which one are you going to buy? Probably the one you can actually steer. It's still almost as fast. You can go really fast, but now you have control over the car. You're not going to crash and die, right? So
Yeah.
that's kind of the way to think about it.
So the chemical signals mostly happen when it's dealing with like the muscles and neurons, or nerve and muscle cells.
Well, pretty much every cell. The chemical are
okay.
everywhere. There's a few electrical ones in our brain.
Which ones are in the brain? Okay, that's what I thought. Electrical is mostly in the brain. Okay.
Yeah.
Okay. Well, I need to study, so thank you for doing this. And I'll try to log on tomorrow at, was it 1 o'clock?
Yeah, I'm gonna do this again at
Okay.
1. So maybe you
Okay.
can, you know, if you don't have to work or something, you can study a little bit in between. I didn't want to do it too late because I want to give you time to study and stuff. But
So you'll be on at 1 o'clock. Oh my gosh, thank you so much.
tomorrow, yeah. And you can always send me emails. I've been at my computer for like 16 hours a day since we started.
Oh my
trying to get all this figured out.
Oh, I know this whole thing sucks.
Yeah.
I'm so not used to online. It's like throwing me off. I don't know how to do like video stuff and
I know, I'm like super hands-on. I'm like, ooh. But thank you so much.
All
We
right.
really
everyone.
appreciate it.
All right. Anybody else got questions?
Yeah, thanks for the questions too. It helps us all.
No. Hello?
Anybody else? Anybody else?
I'm gonna look up.
You see the one
chat.
someone asked about math equations? You got that one?
What math do we have to do?
Yeah, someone was just asking if there's any math on the lecture.
No, you know what? You guys got lucky because that's the day we canceled. Okay, well, I figured it was nice to finish the lecture we're in the middle of, right? And so I had to get rid of some stuff with that day we canceled. So I got rid of some of the equation stuff from this exam.
We appreciate that.
Thank you, coronavirus. But
yeah.
Kind of.
And also the very detailed A&S. And so hopefully you've all seen, and since you're here, I'll mention it, but I'm sure you've seen it. Hopefully all of you have seen the revised PowerPoint and notes, right? That I took out the stuff that you wouldn't have to know. I also posted the revised group problems and put a link to one. I just went over them all. So if you haven't seen that, wouldn't be a bad time to look at that. And if you have any questions about those as well. And as usual, we saw that they weren't every question from last exam, but they were pretty important. So make sure you go over those as well, right? And
Professor, for the revised, I saw the revised group problems. I saw that, but where for the revised like other notes, is that under each topic? Or
no, I put that at the top too, I think.
Okay, so it's just all under COVID. Anything under COVID, is that what you're talking about?
Yeah, so let me look at the modules really quick. Yeah, so it says nervous system changes, right? There's lecture notes, there's a doc file, and then the PowerPoint, and then the PDF of the PowerPoint at the bottom of that. And I put that right at the top of the modules, so you're going to find it right away.
Okay, and that's right under the COVID title, right? I saw the COVID.
Yeah, the COVID edition. Yeah.
Okay, yes.
Thank you.
Thank you. We'll see you at 1 o'clock tomorrow. Thank you.
Hoffman.
Have a good night, everyone.
All right, see you guys. Hang in there.
Okay,
Take care,
you too. Bye.
Bye.
Yes, everyone.